This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me here today, I have Dr. Raquel Fleskes of Dartmouth University. Good morning. How are you?
Good morning.
I'm really excited to have you on. We were chatting a little bit before I hit record, and it turns out we know a lot of the same people and we're interested in a lot of the same things. So everyone's just going to have to tolerate how much we like each other through this episode.
I know, it's always such a small world, and I just love that. Like, academic circles get smaller and smaller and smaller, and it just makes me feel like I know you a little bit more, even though we've just met.
Yeah, no, for sure. So the ways in which we overlap, which I think is actually just a good way to get into the episode, is that you have done quite a bit of work in the colonial Tidewater / the South, specifically in kind of like colonial bioarchaeology circumstances. But you yourself do not do bioarchaeology, you do ancient DNA.
Yes, that's correct. So my pathway into ancient DNA has been through bioarchaeology, but more so thinking about how can ancient DNA add an additional layer to bioarchaeological analyses. So Gabby and I connected because we actually went to the Smithsonian Institution and worked with Dr. Doug Ousley back, I don't know, I was 2014. I don't know when you were. I don't want to time myself too much, but
like 2010 through like 2012. So you came like just a little bit after me.
Yes. And so, Doug Ousley was doing a lot of really wonderful work on the Chesapeake region. And so I grew up in Maryland, grew up going to archaeological sites like Williamsburg and Jamestown and Annapolis. And I went to college at the University of Maryland. And so during that time, this is right when you have, the publication of, the Neanderthal draft genome and the spread of the Neolithic transition and all these really amazing ancient DNA papers. And I just kept thinking like, well, what could we apply to these same methods in ancient DNA to the study of the colonial period? And so what did I do? I just talked with a lot of people who were doing work in colonial period archaeological sites. So like at Avery's Rest or Patuxent Point. And yeah, it kind of made my own way through there.
I think it's worth stopping and mentioning that when people say ancient DNA, I think their minds do jump to like Neanderthals, but that's not necessarily the case. What does ancient DNA actually mean?
Yes. So I get this question quite a lot because people say, well, this is ancient DNA. Ancient DNA should be thousands of years old. And you work in the historic period. So wouldn't this be really historic DNA? And then we get into the weeds of like, well, what does it mean? So
that's what we're here for.
Exactly. So the methods that I use in ancient DNA are really the same methods that you would use to extract DNA from a Neanderthal or from an individual that lived 5,000-6,000 years ago. We're all doing the same thing. It's just the time period is different. Now, because this is kind of a relatively new application of ancient DNA to kind of younger sites, people have argued perhaps we should call this historic DNA. However, the DNA molecule of itself is so degraded, it is like ancient DNA. So, when we pass away, we are buried and our body starts to degrade, our DNA also starts degrading with that. And so the damage patterns that we see in DNA from historic period remains is very similar to the damage patterns seen in ancient individuals. So for that, we're probably going to stick with ancient DNA or aDNA, and maybe stay tuned for a manuscript about historical genomics, which is what I would like to create a subfield in the future.
Yeah, it was actually really cool reading your articles, most of which are easily publicly available, by the way. So thank you.
Mm-hmm.
Because one of the things that really impressed me was, and this is something that I think is very common in the Tidewater, and I haven't really seen it in many other like periods or like areas of research, which is the super collaborative and like integrated nature of the research. So you have, you're doing ancient DNA, but you also are working directly with archaeologists, historians, people who do osteology. I'm sure there's a dental anthropologist in there somewhere because they're always there. But you're like really you're working with a lot of different people to get like a kind of 360 view of any given site that you're at.
Yeah, I think that's why I am a person a scholar that uses ancient DNA, but in the context of anthropology and biological anthropology. I really feel that ancient DNA provides us a strong perspective on things like biological relatedness or population affinity, ancestry. However, that information is only one, I like to call it, way of knowing about the past, right? And so I feel like we can really deepen that interpretation when we talk to osteologists and combine our work with archaeological evidence. And then working in the historic period, we also have the documentary record. And whose narratives are preserved versus whose narratives are not preserved tells us a lot about processes of marginalization. So, what I'm really excited about is thinking about how we interpret lived experience in the archaeological record by using this multi method approach. And so it takes, I remember when I was in graduate school sitting there and I was like, all right, whiteboard. This is what the osteology says. This is what the ancient DNA says. This is what the, archaeological context says. This is what, the archival work says. And I'm like, I got to bring it all together to try to tell the story of the past in a way that I'm trying to account for my own biases, figuring out what the limitations of all these methods are, and then what it can tell us. So it kind of becomes like a little bit of a puzzle piece. And that's never going to be a complete puzzle by any means, but gives us a little bit more of an understanding about who these persons were and the lives that they lived.
Yeah, there was a term I saw in one of your papers that I really liked, which was osteobiography.
Yeah, I love that term. So, osteobiography is a really great, I like to think about it as like a theoretical, but also methodological framework where we are trying to understand the person, right? So, especially in ancient DNA or population genetics, we tend to think at this population level, the movement of people. And that can often create a process of abstraction or of objectification, right? And so, a lot of my work strives to make sure that we're always considering the personhood of the individuals. And the best way I think to do that is to try to think about them as people, like as your grandmother or as your mom or as your uncle or your nephew or somebody you met down the street, right? Their stories are going to be so multifaceted that so the end results of all of our methods need to be as multifaceted as well. So an osteobiography kind of provides that theoretical underpinning for us to kind of try to think expansively about the data that we generate.
Yeah. Yeah. I mean, you can definitely, it's like the whole top down, bottom up.
Exactly.
It's like, let's make it as multifaceted is the best word to use for sure as possible. One other thing that I wanted to pull out that I saw in all of your papers, now that I have read your papers, I'm like, now I understand why you're like friends with Dr. Maria Colon, who we interviewed earlier, was the community engagement aspect of your work. Because a lot of your sites are in populated areas. There are potentially descendant communities. All of the sites are like actively used. And I'm saying sites and not saying what they actually are, which is a lot of your sites are in churches.
Yes. Yeah. Churches or other domestic sites. A lot of the spaces that I work in, because they have to do with human remains, are also emergent spaces. So these are sites that are uncovered during construction. So that uncovering provides an opportunity to do further research about who these individuals are. But going back to the importance of community engagement is that because these sites are living also in a very specific time period, especially in the US context, the historic period. So churches, domestic sites, also sites that are uncovered during construction. I like to think that the US is kind of one large graveyard. We just
Yeah.
build right on top of burials all the time. And so, whether we know it or not, we're likely, in contact with the dead at a lot of the times. But also, community engagement is something I feel really strongly about because of the context through which I work in. So, because of churches, these undiscovered sites that are in the colonial period, we have to think very critically about who is doing the work and who has a stake in doing the work and who needs to be consulted in doing this work. So when we're talking about histories of the colonial period, we have histories of European individuals. We also have histories of indigenous individuals and of African and African descended individuals. And those folks were brought here through the processes of colonialism, whether that is free, forced, or coerced. So in order for us to not replicate the marginalization that has come through the processes of enslavement and colonialism, we also have to radically rethink how we do ancient DNA so that it is accountable to the descendant communities or other communities of care that are impacted by this work. So I like to think about this as I think that we have these legacies of social memory when we work at historic period sites. Either that social memory is because of the historic past, either that's through the direct genealogical connection, but it's also geographic. People live and walk through these sites, or they have a spiritual connection to different sites, or cultural, or religious, or all different ways that people connect back to the dead. And so it is, I think, our responsibility as scientists to make sure that those voices are at the table when we're trying to think about if we wanted to do a study, what kinds of questions should be answered, should we even do the study at all of these things. And I will also note, some people ask me, how does this apply to like deeper time periods that are not in the historic period? Does community engagement matter? And I also argue that it continues to, right? That it is almost as important, because when we don't have those living legacies of social memory, those memories are almost lost or put to the side. So then it becomes the responsibility of scientists to make sure people know about these human remains, or these people who may have faded from memory. But it becomes our responsibility to make sure that people know about them so that we can do engagement and bring awareness to their remains.
Yeah, for sure. I think that I don't know when people will listen to this or when I will put this out, but the Olympics are on right now.
Hmm.
And I live in Oakland, California, and people here are so freaking hyped about Alyssa Liu and her figure skating, which fair, she's incredible.
Yeah.
And like the hometown pride is so strong. And you're probably like, why the heck is she talking about this? It's also true of ancient/historic remains. It's the same thing. If you get the people
Hmm.
who are local into it, they get super excited and it's like becomes part of their community identity, and it helps. Um, before I hit record, we were talking a little bit about how do you get people interested in science, and this is one of the ways, is this community
Right.
engagement. So even if people aren't directly related to whatever remains you found there, people do get like very proud of the things that are local to them.
Yes, I agreed. And I think education is the key here, right? By taking a community and saying, or taking the hands of a community, right? And saying, let's go on a journey together and let's learn about this together. And I want you to be in the driver's seat. What are the things you want to learn about? Here are the things that I can provide answers for. Here are the things I can't provide answers for. Do you understand why I can't do that, or what I can do? That is the key. It's relationship building through education and outreach. And I think it's really it becomes a really fun opportunity for folks when it's done in a way that respects sovereignty of that community, that it really is done with the best interests of trying to build capacity either through education or outreach, or trying to get students or local members involved in the production of the research through whatever means that you can. Yeah, I think it becomes a platform, right? To like bring ancient DNA, which is very classically removed science. There's only a couple dozen ADNA labs in the United States. They're often located really far away from communities. We have an opportunity now to try to democratize that science and make it more accessible.
Yeah. Yes. And then the other thing that is wrapped up into this is that a lot of your sites are they're in cemeteries, and they're in kind of these mixed burial grounds or historically Black cemeteries.
Hmm.
And then one of the things that we have to think about as anthropologists is our history of grabbing remains from people who do not want their remains grabbed, or who we haven't treated, or who have been marginalized throughout history. If you want to learn more about this in terms of like museum collections, there's an episode with Carlina Delicova where we talk about this.
Oh, amazing.
Yeah, pretty extensively. Also, someone I saw referenced in your articles.
All the time. Love her.
She's so nice. But I think that one of the things that is really cool about the research that you're doing is you're kind of like uncovering the experiences, the lived experiences of these people in the past, which is especially important because a lot of the times these are folks that don't get their stuff written down in the historical record in a way that's accessible.
Right.
So actually, I do want to let's get back to like some of the stuff that you can figure out through ancient DNA. What are some of the things that you can learn using ancient DNA?
Yes. Great question. So, ancient DNA, we can learn about population affinity. This is also called genetic ancestry. So, what we're going to do is basically extract that DNA from that archaeological individual. Usually, that DNA is highly fragmented and in really low amounts, but we can get at least 0.1x coverage. So what that means is that if we have on average any place in our genome, we should hopefully have a little tiny bit, zero point 0.1 on average, covered at any position, any sequences, right? And so then we can compare those sequences to a set of reference populations, okay? And we can call what are like we call variants or like markers, or we call them single nucleotide polymorphisms or SNPs. And we basically ask what SNPs are found in this archaeological individual and where are they found in contemporary populations today. And then that is how we would assess affinity. So that individual shares more markers with populations in, let's say, West Africa than in Western Europe. We would then say that person had ancestry to that region of the world. We can also do things like assess genetic sex or chromosomal sex. So if an individual is carrying a Y chromosome, some of those little pieces of DNA that we will extract from that archaeological individual will map onto the human Y chromosome. Okay. And so that essentially, when I say mapping, is we're taking our fragments of our DNA and we're trying to match them anywhere on that human reference genome. So if some of them match to the Y chromosome, then we can say that this individual likely contained a Y chromosome and thus potentially are a genetic male. And then we can, from that information, deduce a Y chromosome haplogroup. So Y chromosome haplogroups is a way of thinking about paternal ancestry, the history of our fathers, right? So if you are a biological male, you will carry that Y chromosome, and so will your biological father and their biological father before that. So we can look at the Y chromosome to look at paternal migration histories. Now, we can also look at something called the mitochondrial DNA. The mitochondrial DNA is a little tiny plasmid, right? It's really great for ancient DNA because it's found in higher copy number. So instead of just having one copy of a mitochondrial DNA, you often will have thousands within a cell. So it's really great when your DNA is really fragmented. You need a lot more copies to be able to efficiently extract it. Now, the mitochondrial DNA is also a lot smaller, and it's only inherited through biological females. So your mitochondrial DNA comes from your biological mother and their biological mother before that. So that tells us our direct maternal ancestry. So we can compare direct maternal and paternal haplogroups. We can see if there are haplogroups that are matching. We can do relatedness estimation. And then we can also look at that autosomal DNA, or the 3.2 billion base pairs, to look at relatedness as well. So thinking about how much DNA we share between two, or is shared between two persons, can be used to construct that. So those are kind of the three main things that I use aDNA for: is relatedness estimation, biological sex estimation, and population affinity.
Yeah, for sure. It's the mitochondrial DNA and when I said MT, that's M, the letter, and the letter T, not MT.
No, that's I heard you.
Not full. just for our listeners, because I was like, because I was like, I don't know. That's a very common abbreviation if you're ever reading articles. mtDNA stands for mitochondrial DNA.
Mm-hmm.
And the Y haplogroup DNA, that stuff is really useful, especially in the deep past, for understanding migration patterns. And one of the things that's really important to colonial Tidewater research in general is migration, because there was a lot of forced migration in terms of slaves being brought over via the Middle Passage to the United States. And then you also have Europeans coming as well.
Mm-hmm.
So you have people who aren't born here who end up here all in a group. Or one of the things that's super interesting about the colonial Tidewater is that you have like a lot of, especially in Maryland, you have like a lot of really interesting religious stuff going on.
Mm-hmm.
Maryland was originally essentially like a Catholic haven colony. This guy named Cecil Calvert.
Yes, the Calvert.
I cannot believe I remembered that. It's a deep pull.
Yeah.
He basically started the colony because the Protestant Revolution was going on in Europe. Catholics were not a group that was super welcome in a lot of places, and so they started Maryland as kind of this place where Catholics could go and thrive. In theory, the Catholic population never got super big, honestly, in the colonies. But a lot of the churches that you work in are Catholic.
Yes. So I got really interested in this. I grew up as a Roman Catholic, so I'd like to think my Catholic school middle school experience was coming back, which is great. Not, on purpose, but I think religion here is a really interesting way of thinking about as a migration driver, right? And it's actually usually very difficult to parse out religion as a motivating factor in migrations using population genetics, because This is not something that's written in our DNA, right? It's very
Hmm.
difficult to tell, but we can make some conclusions when we find individuals buried in a churchyard, right? Or associated with a large church, that they were likely practicing that same religion as that church was, right? Which is why they would likely be buried there. And so we can kind of get at what the influence of religion in affecting migration strategies in these church contexts. So for instance, maybe if I could bridge and talk a little bit about a project down in Tidewater, Virginia. This is the St. Mary's Basilica of the Immaculate Conception. This is the only predominantly African American basilica in the United States. And in fact, it's actually built upon a series of, I think, two churches, a church and then a reconstruction of that church again. So the burials within this church offer a really interesting way of kind of thinking about who is buried at this church, who is welcome to be buried at this church, especially in the context of lots of immigrant communities. And I like to think a little bit about like the social privileging of burial spaces, right? Who is present and who is not. Can we inform economic status, gender status, racial status, and kind of infer what lived experience was through those lines?
Yeah. And one of the things that that you see in cemeteries is that there's a prioritization of people based on how the cemeteries are laid out,
Yes.
right? This is something that the Roy Romano episode covers in New England cemeteries. The way that people think about status is reflected in burial positions. So having a mixed burial ground, even to begin with, is very interesting.
Hmm.
And then seeing where people choose to bury people of African descent in those cemeteries is also says a lot about what's going on in that community.
Yes, exactly. So, in the context of St. Mary's, the church was undergoing some significant renovation back in 2019, right? So they needed to do a lot of structural changes, specifically to the back of the church, but really the entire church got a significant upgrade. It is absolutely beautiful and breathtaking. If you ever find yourself in Norfolk, Virginia, please go to St. Mary's Basilica. It is an incredible church. And if you do go right at the entrance of this church, you'll see a very nice checkered black and white floor. And underneath that floor is actually a honeycomb of burials. There are burials all under this church everywhere. And so when that church was getting renovated, they needed to do some structural changes to basically the choir section up top. And so they had to do a little bit of digging. And so that was going to impact about 6 burials. And so that provided an opportunity that they did need to do burial recovery here. And so the option was raised whether or not the church community wanted to do a more in-depth analysis, like doing osteology of these individuals and potentially ancient DNA along with the archaeological work, which is how I became connected with Dr. Dave Brown at Fairfield Foundation. And so we worked together to do kind of a community engaged study of these individuals that were recovered. Now, what's interesting is that the archaeological context, and please, Dr. Brown, if you are listening to this, please do not, I will do my best. Okay. It's very complicated, right? It's very
Yeah.
complicated. We have some brick lined crypts. Okay. They are a mix of machine made brick and then handmade brick. Okay. And then we also have some wood coffins. There is a drainage tunnel that bisects those brick crypts. It is not organized. You know how I like to think of like classic burial grounds where everybody's nice in a row. It is a little bit interdispersed, okay? And so that's really interesting for burial chronology, right? Because it's, who was buried here first, right? Did those brick-lined crypts were they earlier than the wooden coffins, or at the same time? So what we know from at least the burials that we uncovered is that we have quite a range in the kind of the 1750s to like 1850s, okay? And they kind of span that whole generation of an earlier congregation called St. Patrick's. Okay, so remember earlier I said St. Mary's was built upon another church. Well, that church is St. Patrick's. St. Patrick's was a Catholic church that was predominantly run by Irish and French priests, right? And so they served a lot of Catholics were a lot of immigrants, so a lot of French Catholic, Irish Catholic congregations. And so the question was, well, who is buried here, right? Do we see lots of Irish folks? Do we see French folks? Do we see people who are African descended? We do know that St. Patrick's did serve the Black community as well. Later on, another church called St. Joseph's was formed that was predominantly serving the Black community. But before St. Joseph's, St. Patrick served both congregations. So the contemporary congregation of St. Mary's really wanted to know, well, who was buried here? And then how can that help us think about the history of our congregation? So yeah, I mean, so we did a DNA study. Maybe should I jump right to the results?
Yeah.
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 Okay. I
Yeah.
mean, it's always
Tell
so
us.
fun, right? Well, actually, before I do that, because I always like to call attention to the fact that these projects do not happen overnight. Community-based projects are long-term projects because the process of relationship building takes a lot of time. And so you need to talk to people about what you would like to do. People need to think about it for a while. Then we have to come up with kind of a series of, this is what I would like to do. Are you okay with this? You do a lot of education and outreach around that. And then if you're lucky enough as an ADNA person, you actually get to go into the field to help with burial recovery, which is what we got to do for the St. Mary's Project. And this actually happened in March of 2020. March of 2020, as we all
Damn.
probably can remember with some grimace, right? A little PTSD, was the beginning of the pandemic. So it turns out that pandemic protection measures are almost the same as ancient DNA contamination prevention measures. So we all just wore lots of masks and gloves and washed
That's so handy.
with bleach and kept our DNA from, impacting the DNA of the remains. So I always think, when I think about this project, I think about those fond memories of doing burial excavation in the beginning of the pandemic.
Yeah. It's like everyone remembers where they were in March of 2020.
Yeah, exactly. Where were you? I was in a burial. Like, probably shouldn't have been doing that, but you know what? We had a window.
And took advantage.
Exactly.
Yeah. There's listeners. I'm going to post this article in the show notes, but the article is Archaeogenomic Analysis of 19th Century Burials at St. Mary's Basilica, an intersectional analysis of religion, race, and migration. There's a really, really great graphic of what is going on underneath the church floor. I'm assuming courtesy of David Brown.
Yes, exactly.
They're always like such like interesting, complicated, and it's like got like 8 million colors in it. So if you're colorblind, I think that you can actually use AI to help you figure out where stuff is. AI, I have a lot of feelings about it, but this is actually one of the good uses for it. Or you can ask a friend to help you, like a human friend.
Yes, human friends are good.
Human friends are good.
Yeah, I mean, the archaeological context is crazy, and really that map only shows us just a part of the church. And so when we were doing the burial excavation, we invited Dave Givens, who was at Jamestown Rediscovery at that time, came over and did GPR analyses, and they found that, I mean, under the floor that had already been laid, I mean, it was like burial, 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 burial. So we have to keep that in mind when we're talking about sample size, right? That the sample size of this analysis was only really 5 individuals that we were able to do any osteological analyses of. And then we were only actually able to get DNA from 3 of those individuals. So when we are doing our analysis, we have to keep in mind that this is only a small fraction of what we like to think about as a much larger necropolis, right?
Yeah.
And so the things that we're capturing may only tell us part of the story. I think this is a really important lesson for people who do work in paleogenomics, or maybe anything in biological anthropology, is that, when we see small sample sizes or N of 10, those are, especially with human remains, may only be the people that have remains that have survived to the present day, right? So we're always looking at things through a slight bias or a slight skew. So if we just keep that at the front of our mind, then we're not going to be over-interpreting our results.
For sure. 2 things. GPR is ground penetrating radar, just in case someone doesn't know what that acronym means. They basically just go in and it shows you, well, to me, it just looks like fuzzy dots on a screen. But to people who know how to read it, they're like, oh yeah, that's a body.
It's magic.
It's crazy. I'm like, literally, it looks all the same to me, but amazing. And secondly, one of the things that I wanted to note here is that Norfolk in the 1800s was actually a really cosmopolitan city.
Mm-hmm.
If you've been to Norfolk today, like, I don't know that you wouldn't necessarily imagine it, but like there was a large Jewish population, for example, and the Catholic Church in Norfolk at the time was much more integrated than any of the Protestant churches in general. So you have like these really interesting kind of overlaps with different communities inside Norfolk that you maybe wouldn't expect if you're just like, here's the current group of people here. One thing I also wanted to pull out from your articles is that you look at ancestry groups, but you also do kinship stuff. So with part of the community engagement, one of the things you can do is be like, if you're interested, we can figure out whether the people in here are related to any of the people who currently live here.
Mm-hmm. But with a caveat, though, too, so when we talk to people about genetic relatedness between colonial populations and the past populations, we always have to do so with a line of, every generation, you lose about 50% of that DNA, right? And so if you even go back 14 generations, the amount of DNA that we have overlapping between a living person and an archaeological person is so small. It's tiny, 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 right? And then you combine that with ancient DNA being already very highly fragmented and found in small amounts. We can actually only do relatedness construction reliably about 5 to 6 generations back. After that, it becomes a lot of statistics, and you have to use contemporary populations to estimate the probability of different variant calls. And it becomes a little bit more difficult, especially with African American populations that are admixed between so many different other populations before. So we always preface this with, when I talk to people about it, is that, DNA is one way of knowing and one way of connecting with the past, but this should not trump or be more meaningful than a spiritual connection or a genealogical connection or a geographic connection to these individuals. And if we can show that there is some sort of genetic connection between you and the past, that is wonderful. But the likelihood of that actually happening is really low because of the time difference that is between a 1700 burial population and a living community today. It's about 250 years. Unless we have direct mitochondrial DNA or Y chromosome DNA with genealogical documentation, then we really actually can't say too much. So that's then where, if you take it another step, asking the question, who has genealogical documentation to the past, right? It's mostly folks who are European descendant or been less marginalized in the history of their families, right? Where they're able to have this type of documentation. So it's a little bit of a push and a pull. We like to see what the population genetic history is of that archaeological site and how that compares to contemporary folks, which is what we tried to do with St. Mary's. In fact, we actually found that most of the 3 individuals that we did get DNA from were all of individuals of European descent, right? Which was really interesting. Now, again, keeping in mind, this is only 3 individuals, so there could have been individuals of African descent in other parts of the burial ground. But for the ones that we were able to recover, their ancestries came really actually all from very different areas within Western Europe, which was really interesting. We had some individuals that looked very strongly like populations in Northwestern Europe. So this would be like populations in the UK, which, for this time period sticks, right? This is an English colony, right?
Yeah, makes sense.
Makes sense, right? But for 2 other individuals, they had ancestry related to populations in Spain, which was really interesting and was not expected. And in fact, one of those haplogroups, again, if I don't just jump to the end, but there's so many fun things to talk about with this project. I'm just going to jump to one of my favorite findings. When we're looking at the mitochondrial DNA, when I first did the analyses, one of the haplogroups came back as an L3F. Okay, and haplogroups is a nomenclature system that we use to kind of categorize that genetic variation. Okay, and an L3F, to any anthropological geneticists out there, is a African haplogroup. L's are very characteristic of contemporary populations that live in Africa. They have tons of genetic variants because that goes back to the fact that modern humans evolved in Africa, right? And so they've been in Africa for a longest period of time. So they have more variants. And so when I saw that, I was like, oh, wow, okay, so this individual does maybe potentially have African ancestry. But then when I looked at the autosomal DNA, so that DNA from both our mother and our father, our 3.2 billion base pairs, the signal came back as very European. And I was like, oh my God, what is going on here? This is like a graduate student, or I was a postdoc at the time. A postdoc's worst nightmare. Like, did somebody contaminate the sample? Like, what is happening? Why do I have these completely two different signals? What is going on here? I do the damage estimates, which is what we do in ancient DNA to make sure that the data is authentic. We need to see some damage. It was damaged. We also did contamination estimates. No contamination, very low contamination. So it wasn't me. And I was like, well, what the heck is going on here? Like, why are we seeing this very different ancestry result? And as you do when things are very confusing, you drop it for a while. So I was like, I don't know what's going on here. So we took, about a 6-month break so I could come back to it with fresh eyes. And I did a lot of reading about L3F. And it turns out that there are a couple of branches of L that have back migration. So as much as people are moving out of Africa, right? People are also coming back in to Africa. Africa is a very dynamic landscape, right? There's lots of individual population movements, interregional movements around the Mediterranean is like a highway to connect different people. And so thankfully, somebody back about 10 years ago did a big study on L3F mitochondrial DNAs. So what did I do? Is I downloaded all of that data and I put it and compared our DNA from that individual to that. We did a phylogenetic reconstruction, which basically looks at similarities or differences between all these different comparative individuals from Spain. And what we found is that this lineage of L3F is specifically from the Asturias region of northern Spain, which corresponds with the European and it's really Western European, specifically with Spanish regions, autosomal DNA profile that we found before. I cannot tell you how relieved I was when I met out. It made sense. I was so happy.
My dad did one of those DNA testing things a while ago. And he was really excited to see like all of the different places that his family came from.
Mm-hmm.
It was a single region in Spain. It was 100% a single region in Spain. It wasn't even like, oh, other parts of Spain. No, one.
That's amazing.
So yeah, that could have been my dad if he died 200 years ago.
But it was so interesting though, also like in the context of the site of St. Mary's, like the presence of Spanish associated SNPs and mitochondrial DNAs was like, it also was really surprising. And I think this is where maybe the intervention of aDNA for population history studies in the historic period can really come in. We have a lot of assumptions about who is here and why they came, and a lot of stories that are not written.
Right.
Maybe not major stories, but comprise what is a predominantly all immigrant population, as we should remember in today's moment, all immigrants, right? And so specifically, again, going back to that theme of religion as a driver of migrations, and Spain is a largely Catholic country, and it has a lot of Catholic colonies in the Americas, right? But Virginia was not one of those colonies, right? So Virginia tries to put a little bit of a stronghold in Virginia and North Carolina. They get booted out by the English very early on, and they are down in Florida. And so the presence of a Spanish mitochondrial DNA and Spanish-associated haplotype was very surprising. So it prompted a lot of literature looking into what could have attributed this. There was also one other individual with Spanish-associated ancestry, and some background research indicated that. So we were referring back to Norfolk as being a very populated city, right? It was actually a port city. So many people were coming into Norfolk from so many different areas. And one of those was Haiti. And so it turns out at the turn of when Haiti declared its revolution, a lot of folks left Haiti who were slave owners. And also the enslaved persons who are now freed of themselves fled to different areas. And so one of those areas was Norfolk. So potentially that could be in Haiti was a Spanish colony. That could be one of the reasons, or perhaps it's just like a Catholic religion as a migration. They wanted to be near a Catholic church. We really don't know.
Yeah.
And so again, that's where I always say that this is a part of the puzzle, right? We are seeing this. It is very unanticipated, but I think it just kind of highlights the driver of religion and motivating migration patterns.
For sure. For sure. One of the things that happens with migration today still even is that people like to go where there's other people already there who are like them.
Right.
That's why you end up with like little population centers of different people in different areas.
Exactly.
Also, if people are interested in reading what was going on in Haiti at the time, there was a slave revolt by Toussaint Louverture, the only successful one in history as far as we know. Super, super interesting stuff. And a lot of what's going on in Haiti today is as a result of basically the imperial powers being like, we really need to punish this country so that no one else gets any ideas.
Yeah.
So a lot of like the poverty that you see today is related to the slave revolt that happened
Yep.
in the 1700s. So, or 1800s?
Yeah.
1800s.
1791, I think.
Yeah. Yeah. So yeah. This is just like, it's such like a rich and interesting period in history and such like an interesting area. It's just like the confluence of all of these people together doing different things. It's really cool. I have a very obviously big spot in my heart for the Tidewater.
It's a fun region. We should get more people working there.
Yeah. If I ever go back to biological anthropology, that's where I'll work. But in the meantime, is there anything else that you would like to talk about that you didn't get a chance to talk about?
Oh, gosh. Well, I think maybe if it's okay with you, I would really love to talk about this
Yeah.
new idea that I'm generating right now, which I'm very excited about.
Yeah.
So, because of my work doing community-based methods where I think a lot about how do we democratize the process of doing this, bringing people into the lab, right? We are doing a 3D tour of the lab at the University of Connecticut where I did my postdoc. And we've also done GoPro videos of extracting DNA so we can show those videos to descendant communities. I'm really excited because I'm going to be developing a mobile ancient DNA lab that is to be coming hopefully in the next year, which is great. And so this will be a deployable lab to descendant communities in the United States so that we can do the work directly on site. The archaeological individuals never will need to leave the archaeological site, so they can stay in the custody of the descendant communities or museum institutions. And it's also will be designed to be an education and outreach vehicle. And get this, it's a fun 30-foot Airstream.
Ah!
It's really nice and shiny, and I'm very excited about it.
I'm so thrilled for you. That sounds so freaking cool.
It's going to be really great. So right now we're, wrestling some bureaucratic ties at Dartmouth to try to make sure that purchase order gets through. But we're going to be working with the Thayer School of Engineering to help build out this lab. It'll be a one-chambered lab with an anteroom with some positive pressure HEPA filtered air. So I'm really excited because I think it's really, again, it's going to change the positionality of how we do ADNA, where we're going to be able to involve the descendant community more directly. Instead of having the descendant community have to come to us, we can go to the descendant community.
That is really cool. I'm so excited, and I'm excited to hear how it turns out and like follow along on your journey. It's gonna have to you're gonna have to name the trailer.
I know.
Like you're gonna
right
have
now
to.
It's just called Mobile Ancient DNA Lab. But we should probably come up with a really fun name. Like, I don't know. We'll have to think about it. Maybe your listeners can come up with some fun names for us.
I know. I'm just like I've been on a really big Scooby Doo kick, so I'm like, obviously.
Oh, that would be fun.
Like...
Yeah, I want to like put like a cool little like double helix on the side of it. You know what I mean? Like, we're gonna
for sure.
get a little tracker. You go online, see where it is, where it's deployed, and maybe we do some like cool live streaming for people to see what we're doing in the lab. But
Dartmouth absolutely has an art department, right? Like they can definitely.
Oh my God, we have so many motivated like undergrads here. People are really excited. So, and I'm super excited. So
that is so sick. I'm so excited for you.
It's gonna be fun. Maybe we'll come rolling around next year to the AABAs with the mobile lab. Might be really fun.
I think next year it'll be West Coast, so you can park it in my garage, I guess.
That would be a very long drive. Maybe next time it's in the East Coast. We'll take it down.
Maybe next time it's on the East Coast. Well, that's really cool. I'm like trying to remember. Oh, right. The rest of the interview. Yes. Question for you. If people want to learn about ancient DNA in general, what's a good starting spot for them? Or if they want to learn about your research in particular, where's a good place for them to start?
So I would probably, rather selfishly, if you want to learn about my work, go to my project website, which is raquelfleskes.com. This is a byproduct of the job search era. Maybe it eventually will become the historical genomics lab as this lab matures. But we have all of our papers listed there. If you're interested in learning about ancient DNA, there's some really, we're starting to develop some online platforms. If you go to jamestownrediscovery.org, our 3D tour of the Yukon Ancient DNA Lab is live there. And so you can click on that link. We're just going to be submitting our publication pretty soon. But that gives you the 3D spatial tour, but then you can watch different videos about the processes of doing the lab work. So you can see us like doing a library prep or DNA extraction. You can click on a little equipment and you can kind of get a feel for what the space is and how it's used.
Yeah, the Jamestown Rediscovery website in general is really good. They put a lot of work into this. So for a long time, people thought that Jamestown was lost to like the sands of time.
Mm-hmm.
And they actually built like a fake Jamestown near where they thought the real Jamestown was, as like part of like the Jamestown anniversary celebration. I think the Queen of England came to come see it. And then like, I want to say like a year or two later, they found the actual site of the real Jamestown, like a mile up the road.
Yep. Which you also check out if you're in Tidewater, Virginia. It's very cool. So you can see the reconstructed, but then you can also see the archaeological site too, which is really fun.
Yeah. And the archaeological work that they've done there has been incredible. And this is really close to colonial Williamsburg. So if you want to have a really fun, like historical bioarchaeological vacation.
Exactly. Yeah. We're actually
to Southern Virginia.
Doing the DNA of the 1607 burial ground right now, which is really exciting. I know. So we've gotten some genomes. The DNA is, as you would expect from individuals that were partially eroded by the James River, not wonderful, but we do have DNA from them, which is really exciting. So please stay tuned. I'm trying to get that published like as we speak.
That's so cool. No, that's really exciting. And for listeners who aren't as familiar with U.S. history, 1607, that was the first like wave of settlement into Virginia from England. So that's like, it's like the earliest colonists. Besides Roanoke.
Yeah. So, I mean, they're the first folks to come over and pass away at Jamestown. So literally, I like to think about it. If this is the first permanent English settlement, those are literally the beginning.
Yeah.
Right. And so I'm really interested in also understanding how population genetic diversity changes over time, which is the really nerdy part of this work, is like, how does genes change over time in population genetics, but a little bit less fun to talk about on a podcast.
Dude, no, that's got to be so interesting because like so many people came and died so quickly.
Yes.
And there was like all of these waves. I'm sure that is going to be really fascinating. And then of course, England was like, well, we need to send some women over there to like make people stay. Like the whole thing is, okay, sorry, no, I gotta focus. gotta focus. What are you reading for work right now?
I'm reading, so me and my lab group, and I read The Trouble with Ancient DNA by Anna Colleen, which is a new book that came out, I think a couple months ago or last year. And it's kind of talks about the criticisms of ancient DNA as kind of being a celebrity science or the powers of narrative, right? And how ADNA needs to do a little bit of a better job holding space for all the assumptions that is embedded in the statistical analyses of our work and what we think about as a population and how we depict migration and movements. And so it was a good read. I think there's a lot of more work to do in our field as we continue to mature.
Yeah. I mean, the work is never done. And final question, what are you consuming for fun right now?
Oh, gosh. I am currently working my way through the X-Files, of which I have never watched before. But I love sci-fi. I'm a big sci-fi reader. So, oh my God, like to find a show that just tickles the little sci-fi just a little bit, but that also crime show and also early 90s, late 2000s nostalgia. Oh, I've been having a wonderful time. And, it's an early TV show. So there are 24 episodes in one season. So it's amazing. So I have 12 seasons of 24 episodes. I'm going to be watching this for the rest of my life. It's going to be great.
It's so funny. No, first of all, the fashion on that show is incredible.
Amazing.
The writing on the show is so good.
Great.
Third, I like that you're just watching serious Scooby-Doo.
Yeah, that's exactly what it is. It's adult Scooby-Doo.
Yeah.
It's been very fun.
Oh my gosh. Now I'm like, maybe I should go back and watch the X-Files.
This is a call to all of your listeners to go back and watch the X-Files.
Well, I'll finish Scooby-Doo soon and then we're still switching. Okay. Well, thank you so much for joining us and I hope you have a great day.
Thanks so much.
Good luck with your lab.
Thank you.
Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.